Good morning, good afternoon, good evening, everybody, wherever you are. Welcome back to the ICH Journal Club, the International Academy for Clinical Hematology. This is our first session after the summer break. I'm Mohamad Mohty from the Sorbonne University and Saint-Antoine Hospital in Paris, in France. And for today's journal club, the steering committee has selected this recently published paper in Annals of Hematology. And you can read the title here, Durable Outcomes with Manageable Safety Leading to Prolonged Survival with tagraxofusp for treatment-naive patients with blastic plasmacytoid dendritic cell neoplasm, real-world results from a European named patient program by Dr. Herling, Angelucci, and colleagues. Of course, I will not expand a lot on BPDCN, which we have addressed in different sessions in the last couple of years, but definitely this is an orphan hematologic malignancy with an extremely poor prognostic, very aggressive clinical presentation. And actually, and we'll discuss this with our guest today, the major advance in this disease was about the advent of CD123 targeted drug, namely tagraxofusp. And initially, this drug, which is great innovation for this terrible disease, was approved and used in the relapse refractory setting. However, the European named patient program that started in 2019 enabled patients with BPDCN with an opportunity to receive tagraxofusp as the first targeted drug in this aggressive orphan disease ahead of commercial availability. And of course, this named patient program represented and still represent a valuable and great opportunity to guide clinical practice and glean some new information beyond clinical trial evidence. And this is exactly what the co-author here did. They gathered the data of 26 adults, patients with treatment-naive BPDCN who were treated frontline with tagraxofusp. And the primary endpoint was about complete response rate, and we know that in this disease, actually achieving CR is a surrogate marker for a better outcome, but also for being able to be bridged to transplant. And we can see the characteristics of the patient. They are really very advanced patient, median age 68 up to 87. You can see the ECOG performance status prior to therapy was 2 in 14%, 3 patient, 1 in 43%. And we have the usual organ involvement, number 1, skin and bone marrow, followed by lymph nodes, spleen, and blood. But we have also 21% of the patient, 3 out of 14, who had a CNS involvement. And despite these very bad features, when you look to the whole cohort, actually, the CR rate was 65%, which is quite unprecedented. And you had a 90% overall response rate. And if you look carefully to the transplanted patients, those who were able to proceed to transplant, the results are very elegant with 75% CR and obviously 92% overall response rate. And you can see here the overall survival probability. Actually, you can appreciate it's a decent follow-up, but definitely the outcome of these patients is improved without the use of the classical conventional chemotherapy. So clearly, these results, this study, in my opinion, but we'll discuss this with our expert, establishes tagraxofusp in a real-world setting as the first-line treatment of choice for a broad spectrum of patients with BPDCN. And to discuss this paper, I'm very honored and pleased to welcome Dr. Emanuele Angelucci from Italy, who is very well known to all of us, an expert in this field. And he is the co-author of this paper. Unfortunately, Dr. Herling, who was supposed to join us, had a last-minute emergency and could not join, but I'm sure we'll have a wonderful discussion with Emanuele. Emanuele, thank you for joining. How are you today?
Fine. Thank you. Thank you, Mohamad. I have to say that the honor is mine to be part of this your prestigious journal club. I'm very happy every time to collaborate with you, with your unit, particularly in this journal club.
Thank you very much. It's a pleasure having you. And of course, I would like to remind all of you that this is a live educational activity. So please don't hesitate to share your questions, your comments, whatever suggestions you have to our panelist, to Dr. Angelucci. So Emanuele, before going into the details of the results from this beautiful study, because also only 26 patients were included, but this is a lot when it comes to an orphan disease. So what are, before tagraxofusp, usually what do we use? What is the first-line treatment in these patients? And why do we need alternatives?
Marco is here. Marco is here.
Oh, I can see Marco. How are you, Dr. Herling?
Hi, great.
Okay, welcome.
Sorry for being late.
Yeah, we know. I already excused you to the audience, but we're glad having you. So the first question, it was about only the first question, and we didn't even get the answer yet. It was to Emanuele, because this is how it works in the journal club. So the question is about the before the advent of tagraxofusp, what was the standard of care in the frontline setting for these BPDCN patients, and why something else was needed?
Okay, that's the answer is very simple. There was no standard of care. So, and was any center is better to treat this aggressive neoplasm disease, and because of the few number, the limited experience, and there are in the literature very few retrospective studies with several different regimens used as first-line treatment. And then I can told to the colleagues in Italy that there's a story that had been audited by the Italian regulatory authority in when they evaluated tagraxofusp. And the major problem is that it was not possible to compare with the standard of care because there was no standard of care. So it was very difficult for them, for those bureaucrats, to look at the clinical benefit. So the strange figure is that even if BPDCN is a myeloid neoplasm, in a retrospective way, it was better treated with lymphoid acute lymphoid treatment. There was a difficulty to it because now we can understand, because this is typically dendritic cells, neoplasm, probably some ALL or lymphoma-like treatment are more effective than AML, like treatment. Nevertheless, there was no standard of care. Any center has its own protocol, but when you treat one patient every 2, 3 years, 4 years, it's very difficult to have a consistent experience and to try evidence. So the value, the scientific value, the innovative value, one of the value of this new drug is to have a uniform experience worldwide, at least in several centers, permitting us to establish which could be the correct treatment for this disease.
Thank you, Emanuele. So my question to you, Marco, and welcome, and thank you for joining us. I know you had a busy day and you have many commitments this afternoon, so I'm very grateful for your time. So you led this study, and can you just remind us why these patients received actually tagraxofusp frontline, and whether the features of these patients reflect actually the real life, the classical standard patient we would see in this disease?
Yes. Yes. I might just pick up where Emanuele left, and I wanted to emphasize this. The difficulties with this disease are inborn. When you don't know whether it is lymphoid or myeloid, then you don't know really how to treat it. And this is one of the predicaments where we started out from. Tagraxofusp, US approval, and that study came about in a time when we kind of settled for, well, maybe ALL-like treatment is the best based on retrospective data, but we were far from calling it standard because it had never been approved in a clinical trial or in any kind of fashion. It was just retrospective data, multicentric, and we kind of felt aggressive ALL-like regimens seem to be conferring the highest CR rates. However, the patient must go to transplant. And as hematologists, we know that even though among the choices, CHOP or AML or ALL-like, the three major, let's say, options that we had, the most aggressive seemed to confer the highest CR rates, but also came at the price of a high dropout rate. For anybody who treats ALL patients knows that it's not 100% of the patients that we bring to an allo, even if initially intended. We lose some of them due to hematotoxicity or indirectly due to infections, fungal and bacteremia and otherwise. So there was definitely a clinical need, definitely for something that's more targeted. And by the time that study kind of ensued, it was not planned as a study. It was basically the company saying, we make that compound available in Europe, knowing that it's already approved by the FDA. Let's try to close that window for the sake of the patients, because we believe in the drug. The drug seems to be efficient. The drug seems to be safe. Let's let the European physicians decide whether they want to give it in the first line or whether they want to apply it to a patient in the second or higher line. So basically, you filed for that individual patient. I need in an early access program tagraxofusp, and I want to give it to my patient. And this was basically randomly spread all over the centers that had access to that program. You could pick it for your patient in first line, or you could pick it for your patient in higher lines. So in this paper that we are talking about today summarizes, without the primary intention of doing a study, but it summarizes retrospectively the data across Europe of these patients that were treated in first line. Now, that is your question, is this cohort representative of the all-comers that are diagnosed with BPDCN? I would say yes, even though each one of us would agree of the shortcomings of a study that wasn't primarily intended as a study, basically a retrospective analysis where biases are being potentially introduced by, for example, physicians' choice, center-specific aspects, and all these things, and the retrospective nature. However, you have this also in a prospective clinical trial. You can introduce biases because you might have inclusion-exclusion criteria that are not real-world-like. I think this scenario here of that paper is closer to the real world than the prospective pivotal trial that was published in the New England Journal that led to the approval. Fortunately, we did not divert in patient characteristics too much from it, and I think it's now even underscoring the efficacy and safety of the product in the first line. And it, we can discuss about this probably later. It brings about certain advantages compared to the conventional chemo. So yes, I think it is a representative cohort, although it's retrospective, but fortunately it is representative. It confirms the results of the pivotal trial, and I think, in my judgment, it is more real world than the actual pivotal trial.
Thank you. And I'm glad you're bringing the issue of feasibility because, and maybe here, Emanuele, you can jump in. I recall from one of our French national multi-center studies with chemo, actually almost 40%, 38% of the patient with BPDCN, and that was monitored prospectively, could not actually proceed to or complete whatever chemo they were planned to receive. And here my question to you, Emanuele, what is your feeling about the feasibility, the ease of administration, and we'll talk on a broader perspective about the safety profile, which is quite excellent, I think, compared to what you would expect from intensive chemotherapy in an older, frail, and population with bad shape, I would say.
Okay, so I believe that on this point of view, tagraxofusp is an important step forward in the treatment. Basically, we have two categories of patients: eligible for treatment and patient not eligible for transplantation. In the first case, I think that tagraxofusp represents an ideal, let me say, bridge to transplant in complete remission, because also in a large percentage of patients you are seeing is able to induce a complete remission in 2, 3, 4 months. And this permits the patient to go to transplant without the toxicity of previous chemotherapy. And this is a major advantage. Second, as any drug, even tagraxofusp has its own adverse events, but these are different from classical chemotherapy. Particularly for tagraxofusp, the most important adverse event is the capillary leak syndrome. And one of the major points of the paper authored by my friend Marco is this: in this European extended access program, no patient died by capillary leak syndrome, while in the registrative paper program by the MD Anderson, there would be few patients, 2, 3, if I remember well, who died by capillary leak syndrome. This means that European physicians, maybe also American, of course, are being able to rapidly recognize this complication and successfully treat. And so this is really an important point, because as you know, you are a transplanter, so you have seen capillary leak syndrome in transplanted patients, and for this complication, what is fundamental is the rapid, immediate recognition complication and immediate treatment, and this really makes the difference. And then this European experience clearly demonstrated that all the centers were able to have this prompt treatment of the complication. For what concern the other group of patients, elderly patients not eligible to transplant, clearly there's a not chemotherapy approach, not intensive chemotherapy approach is preferable, and they can at the same time induce a rapid complete remission and with the same complication rate that the younger patient, so the complications are the same as CLS. Of course, without a transplant approach, this disease is likely to relapse. And then therefore, in the future, even for this category of patient, probably we need some sort of combination therapy, putting together this innovative approach with some form of chemotherapy or some other form of therapy. Maybe now is very famous venetoclax. I know that in France you have a pilot study with the association of tagraxofusp and venetoclax. Anyway, the major advantage, one of the major advantages is the feasibility and the safety of this new innovative product.
Thank you, Emanuele. So this is really very reassuring when it comes to the safety. Now I'll switch with Marco to the efficacy, because obviously the goal in these patients is truly to achieve CR, because CR is a sort of a mandatory step to either proceed to allotransplant or even allow them to enjoy longer remission and eventually a better outcome, better survival. So Marco, how do you assess the response rate that were achieved in these patients, and how do they compare to the historical data? Because obviously, if we want to switch our treatment algorithm from the conventional mixed bag of chemotherapy to a targeted therapy, still you need to show that this targeted therapy is really worth it in terms of responses.
Yeah, that is actually the to a clinician probably most interesting question. What is the actual efficacy and how does it compare? Let's call this one here a real world experience. How does it compare to the pivotal prospective trial data? And in the overall population with tagraxofusp given at on average 2 to 4 cycles as a single agent, we observed an overall response rate of about 90%, of which 65 were complete responses, and the other 25 partial responses. That being said alone is already a convincing argument to say this is an efficacious drug. Given what Emanuele said, it is also a safe and feasible drug. And given the fact that we thereby, by something that is much milder than an aggressive ALL-like regimen, we automatically broaden access for a broader, a wider population, ACOG-wise, SORO score-wise, to the compound to be systemically treated, because not everybody would have been eligible for an ALL-like protocol, but now is eligible for tagraxofusp. And therefore, we give a broader spectrum from ACOG 0 to 1 to 2, the chance to get the drug and get to the transplant, because it not only matters in which state of remission you are before transplant, it also matters in which general condition you are before transplant. People tend, by looking at these curves of efficacy, tend to forget that, right? What is the use of a CR if I'm no longer transplant eligible? I cannot sustain that treatment success. And therefore, to me, it always, if I'm asked, it makes a big difference whether my CR is achieved after consolidation 1 of a German ALL-like intensive ALL-like protocol, or whether a CR is achieved after 2 cycles of tagraxofusp. It's a big difference. And there's no data that would indicate that the relapse rate is particularly higher by either one or lower by either one, right? So it matters how I get to the transplant procedure. And you should never compare two independent in their design different studies. But if I'm allowed to do so, the CR and OR rates in this first-line setting of real-world treatments was not inferior, at least equal or even slightly better than in the pivotal trial of treatment-naive patients. And the rates of patients successfully bridged to transplant, about half of them, was also comparable to the rates observed with respect to inclusion-exclusion criteria, much stricter prospective pivotal trial. So therefore, I'm very comforted to observe that the real-world data of that manuscript, of that paper, hold up with the results of the prospective clinical trial data. And it's also reassuring to see when we analyzed the transplanted patients only, that the benefit added by the transplant in terms of overall survival does not disappear, right? So tagraxofusp is a way to transplant that doesn't make the transplant worse. Also important. We have seen drugs that seem to, biologicals, for example, that let's think about CD22 agents, for example, where we had to learn in the context of a transplant that they bring about other problems, VOD and others, right? So those things were not an issue. So we have something that brings about half of the patients looked at the overall population successfully to the transplant, and the transplant is not in its efficacy and toxicity not negatively affected by the compound.
One quick question, Marco, and this is a question from Dr. Dereen, whether you would proceed to transplant only in CR, or you would go with a lower response if it's a young patient in good clinical shape. How would you approach? Would you really hit hard to achieve CR, or you can proceed? And maybe that brings me to another question from the audience about the use of TAG maintenance after allo, especially in those patients who had some residual disease.
Let's answer the second question first. There is maintenance strategies at MD Anderson. There's a trial where they look at that. I'm not aware of any data, but the approval text, the indication in the label, it's not included. So it's in the EU, it is for first-line treatment only. We cannot use it as a maintenance strategy after an allo. But there's this MD Anderson trial that addresses this, whether tagraxofusp consolidates the responses in a way that, if given as a maintenance after allo, relapse rates are even more reduced. The other question is a bit more difficult to answer because it depends how much more can my patient handle. Let's start with the assumption that the better the response, the better the outcome of the allo will be. We know that the allo works apparently better if it is not reduced conditioning. It seems to be a myeloablative. That's one thing you need to make sure. And it probably should include some form of TBI. That's what the data suggest as well. Now, does that justify to be happy with the partial remission? I would say no. I would still try to say my patient has not suffered major toxicity. I'm at cycle 2, tagraxofusp. I do know that some responses come even after 4 cycles. I would probably give 1 or 2 more cycles or switch to a short course of chemo because that disease per se is chemosensitive. It just has this habit of relapsing all the time or very often. So it's not for the lack of primary responses. So if I am in only a PR after 2 cycles of tagraxofusp, I would probably say, let's either do 2 more rounds of tagraxofusp or switch to a short course of intensive chemo and then go to transplant, hoping to accomplish a CR because usually that disease is chemosensitive.
Excellent. Thank you very much. Very clear. Emanuele?
Okay, completely agree with Marco. Complete remission before transplantation is essential. I have a patient, very young patient, with CR must described the residual disease, a 16 years old patient with residual disease after 2 cycles of tagraxofusp. I treated with a 1 short course of intensive chemotherapy immediately after allo transplantation. So this is really, really important. But, and I support the inclusion of bridging intensive chemotherapy if complete remission has not been achieved. But there is another consideration on this point. And then if you achieve a complete remission, you must go to transplantation as soon as possible. And in my opinion, in this particular situation, particularly for young patients, time is more important than compatibility. So this is a classical situation if you lose time with the search for an unrelated donor, maybe an Afro donor would be better because timing is essential, a complete remission is essential. I also support the use of myeloablative conditioning, in particular with total body radiation. There is a paper, I don't remember the title, supporting a cumulative effect of a particular sensitivity of BPDN cells to radiation. And they support the use of total body radiation as preparative regimen. As this is similar to conventional therapies, this disease, although the disease is a myeloid disease, but this disease seems to have a better response with acute lymphoid leukemia intensive treatment than with other treatment.
Excellent. Thank you very much. So my last question is to you, Marco, before concluding. What is or what are now the next steps? So it looks like that the tagraxofusp now is becoming a well-established frontline treatment. So we're moving into earlier lines of therapy, excellent safety profile, high level of efficacy, and allowing to bridge many patients to transplant. So what are the next steps if we want to cure more patients?
Yeah, so that's probably a multi-layer answer, and I very likely forget some of the aspects. Maybe Emanuele will then add what I forget. I think open questions scientifically, there are a lot, and I'm not sure whether the company's interest align with those. But I would want to see that drug also being available to us in second line. There are countries, there are physicians that prefer or only have available as first line holy chemo. Why should they be punished for not having access to tagraxofusp, even if it's only in second line? Because we know it also works there. So you can have a patient that seems to relapse or be refractory to intensive chemotherapy that you could still rescue, if you will, and even bring to transplant with tagraxofusp as second line. So I would wish to see an approval in second line to broaden my treatment armamentarium, right? Then how about those patients where you already know you cannot bring them to transplant because they are too comorbid or too old? The elderly patients that are right up front not allo eligible. We know if you're lucky, you are a responder and you are a sustained responder under permanent tagraxofusp. Those 2, 3, 4 lanes in the swimmer's plots that you always see on top, right? But that's why I use the term being lucky. The majority of these patients eventually will then be refractory or relapse after a tagraxofusp response initially. And then you are pretty much left alone with either back to chemo, mild chemotherapy, or can we improve tagraxofusp efficacy by combining it with other agents that we know are tolerated in the fraction of patients? Let's compare them to those where we would give Vidaza venetoclax in AML. The elderly fraction where you think, well, maybe we get them to allo, but most likely not. But let's start with the palliative Vidaza venetoclax. And I would say if you add as a third agent tagraxofusp, or if you just used tagraxofusp plus venetoclax as a combination, being tolerable for an elderly subset of patients that you don't want to transplant, that's something I would like to see company taking the initiative and addressing those kinds of questions. And of course, there are issues around CNS involvement, where we know tagraxofusp doesn't go into that compartment, but we have a high incidence of CNS disease. How do we address this? And then, of course, tagraxofusp, allo, and then the relapse. What do we do then, right? What are our options? So we need more combinations with tagraxofusp, and I think we also need more new agents.
Wonderful. Thank you, Dr. Angelucci. Thank you, Dr. Herling. Thank you all for a lively restart of the IACH Journal Club after the summer break. Today's paper adds, I think, meaningful evidence in one of the rarest and most challenging hematologic malignancies, namely BPDCN, by focusing on the frontline CD123 targeted therapy with tagraxofusp. I'm very grateful to you, Emanuele and Marco, for your involvement and for advancing and improving the natural history of this disease. Thank you all for the very sharp questions. It is such a pleasure to kick off the new season with such energy, and we look forward to see you at our next ICH activity and of course a journal club. Until then, please keep sharing cases, please keep enrolling patients on trials, and pushing the field forward to cure more and more patients. Thank you very much and take care.
Thank you. Bye-bye.
Likewise. Thank you.